Hi everyone, I'm Pulkit, sixth year PhD student at Penn. And today I'll talk about our work on postmortem brain analysis at 7 Tesla to understand Alzheimer's and related dementia. Just to start with the acknowledgement slide, I'm in Paul Yuskiewicz group, and I just want to thank all the tissue donors and their families, and also the following NIH grants which supported this work. So we know that comorbidity of Alzheimer's and related dementia and conventional in vivo biomarkers makes it really difficult for clinicians to determine to what extent cognitive decline in individual patients is driven by AD versus other factors. And therefore, current in vivo biomarkers are unable to reliably detect non-AD pathologies from normal aging. Therefore, there is a need to identify and obtain structural measurements from AD-specific regions so that then we can derive in vivo biomarkers that can detect and quantify mixed pathology. So we believe at Penn that a joint analysis of ex vivo MRI and histopathology could help link changes in brain morphometry with underlying pathological processes in AD and related dementias. And this would eventually help validate in vivo biomarkers that consider pathologic heterogeneity in clinical trials. Right? So our protocol at Penn is usually we have two brain hemispheres. One of the brain hemisphere is imaged at 7 Tesla, and the other hemisphere is used for histopathology. And also these donors were patients at Penn Memory Clinic, so we also have longitudinal antemortem T1, T2, flare, diffusion, and in some cases fMRI sessions as well. So much of the focus of the lab is in the medial temporal lobe and hippocampus, hippocampal subfields. So in the previous slide I showed that we imaged the whole hemisphere, but also per hemisphere or for every brain specimen, we take out the MTL and image that at 9.4 T. And so that's all ongoing work in the lab. And I'm not going to talk about the MTL stuff today, but stuff that I am working on, which is whole hemisphere parcellation and registration. So just to give you an idea, we have over 200 postmortem MRIs of these donors which span Alzheimer's continuum, Lewy body dementia, FTLD, both due to tau and TDP, and all these other different diagnoses categorized into primary and secondary. So in particular, for every brain specimen, we have three different modalities: a T2-weighted image at 300 microns, a KISS scan at 500 microns, and a T2 star flash sequence at 160 microns. And remember, additionally, we also have all the antemortem data for all these patients. Right? But to start any neuroimaging study, you need segmentations, parcellations, and you need a good registration method. But postmortem imaging is really tough, which I don't have to tell you, due to the following challenges, and some of the challenges are specific to our data sets. So we have greater heterogeneity in scanning protocols. There is just greater textual complexity between scans. Again, these are severely demented patients, so every brain scan looks very different from each other. There are large collapsed ventricles in many of these scans. Then there are flattened edges due to the fixation process. And there's also profound imaging artifacts, which was a limitation of our previous coil, where we have signal dropout in anterior and posterior part of the brain. Moreover, since the data set is part of larger studies, we have these sampling cuts because that part of the tissue was used in some other project. So we need to be able to fill up those gaps as well to have a nice parcellation method. And in particular, it's just challenging to study 70 postmortem whole hemisphere because there aren't really any methods out there to do this kind of stuff. Much of the focus is on hippocampal subfields or specific part of the region of the brain, but to do a whole hemisphere registration between postmortem MRIs or parcellation, there's just not much except this new paper which came out from UCL group called NextBrain. So over the last 3 years, what I've been working on in the group is to develop a whole hemisphere parcellation and segmentation method, just like how FreeSuffer does for numerous atlases in native space, and then validate the methods using structure pathology associations based on the contralateral and also ipsilateral histopathology data, and then build vertex-wise analysis tools so that we can perform group-wise and vertex-wise studies across the whole hemisphere, just like how we do it for in vivo. Right? And all the work in this talk is openly available at our in a Docker container and Singularity container, and also in this new Python package called Purple MRI. So just to give brief overview of the segmentation protocol, we combine deep learning methods and classic surface-based modeling techniques to obtain, first we obtain 10 different regions in the brain: gray matter, cortical gray matter, white matter hyperintensities, the 4 main subcortical structures, and also not shown in the figure, but core segmentation of the MTL, the ventricles, and the corpus callosum. So we do extensive validation of NN unit. We gather a lot of different manual patches, do a lot of different training, and then do extensive validation. And once we have these 10 labels on every specimen, then we take this T2-weighted MRI. We obtain those 10 labels using our deep learning pipeline. So that's stage 1. And then we adapt FreeSuffer kind of techniques of surface modeling to get whole brain parcellations for any atlas. But this time, instead of putting everything to 1 mm space, we are doing that in native 300 microns resolution. So we take T2-weighted MRI, we have this initial volumetric segmentation, and now we just work on this volumetric segmentation. So like yesterday, how Lindsay adapted the FreeSuffer pipeline, and this is in a very similar way, we adapted the same pipeline as well. So we take that initial volumetric segmentation, get the white matter mass from there, tessellate the surface, do topological correction, inflate it, do a spherical projection, bring in the template, and then do spherical registration based on the spherical curvature atlas. And then after that, you easily get your PL surface and the white matter surface. And then finally, you can get whole brain surface parcellations at native subject space resolution. So the deep learning pipeline takes 5 minutes for inference, and this pipeline takes around 50 minutes to 60 minutes, depending upon your computing power. And you don't need any GPU for this. Yeah, so these are some examples of how these images look like on a DKT atlas. And then this is another example. And then we also compared our methods to SynSeg and NextBrain, and we do see that those other methods fail, but I think that's primarily due to the registration between their template and our images is just not happening correctly. And then nothing really stops us from getting any atlas out there in the world in this native space, because we have that registration warp and we can just apply that, and it just takes a few seconds to get any atlas in native subject space. So we did this across all the 200 postmortem MRI scans, and then we also generalized to other imaging sequences that I was talking about earlier, the T2 star flash sequence at 160 microns. So for the first part of the segmentation, we obtain all those 10 labels, and in addition to that, we have in our newly updated model, we also acquire additional labels of subthalamic nucleus, hypothalamus, optic chiasm, anterior commissure, and fornix. And now moving on to the second part of the talk, I'll speed up a bit. Now once we have these parcellations in all these 3 different kinds of modalities, what we do is we get histopathology data from the contralateral tissue so that we can first validate our parcellation schemes and also then to understand how AD progresses in different parts of the region of the brain. To do histopathology, what we do is basically we come up with these 18 to 20 locations in the brain which are implicated in dementia, and we take those sampling cuts that I was talking about earlier and stain those tissues for these five different kinds of pathologies: amyloid, tau, alpha-synuclein, TDP-43, and neuronal loss density. And then based on a semi-quantitative score, we manually rate each subject from 0 to 3, going from none or rare, mild, moderate, and severe. Right? Now I'll just talk about some of the quantitative stuff that we can do with all this data. So first thing is we develop an in-house pipeline to validate our deep learning method, the initial volumetric segmentation. So what we do is at each of those 18 locations that I was talking about, we take the automated segmentation and also a small manual segmentation, and then we just do some simple correlations to get compute dice overlap and do correlations to see how well they do. And then the second part of the talk, I was talking about whole brain parcellation. So once we have those whole brain parcellations, what you can really do is, for any atlas, you can go into a given region and just compute your mean thickness versus each of the pathology ratings in a particular ROI. And we see that in darker purple color, we see that it's more negatively correlated, which is as expected. More atrophy, more severe the rating, right? So this is just some kind of validation to see if the parcellations are doing the right job, because in reality, it's just hard to get manual data on such a large cohort. And then this is something really new and we are excited about, is that once we have all these parcellations and thickness maps in native space for all these 200 postmortem scans, we can just swap that to FS average space, and then in that template space, now you can do vertex-wise and point-wise analysis, just like how you do for your fMRI studies and your T1, T2 studies in vivo. So shown here is all the thickness maps mapped into the template space, FS average 40, and now we are just doing a generalized linear model where we fit a GLM between vertex-wise thickness and the corresponding MTL pathology, or the other stages, amyloid, PROC, and CDAT scores. And what we see here is the T-statistics map. The hotter the color, the more better the correlation, and in those black clusters, you can see that those regions which are implicated in AD are the ones which come out to be statistically significant, the entorhinal region and the MTL. So this is another part of the tool which now it gives us the ability to do group-wise analysis in postmortem. Finally, just want to close off with what we can really do with all these segmentation stuff, and parcellation is like nothing is stopping us to explore any region in the brain and study how subcortical structures and the other structures that I was talking about, they are implicated in AD and also FTD spectrum. And white matter hyperintensities, this is something which we are seriously looking at, is compare in vivo flare and these ex vivo white matter hyperintensities, and then see how those are spread throughout the brain in periventricular spaces and also anterior and posterior part of the brain. And last but not the least, another project that we are doing is looking at layer segmentation across the whole brain and then mapping that information with the neuropathology, the histopathology that we have. And specifically, we are looking at the FTD spectrum to see how TDP and tau differentially affects FTLD. Finally, this is the last slide, and what I want to show is that, okay, we have achieved segmentation and parcellation. The last step, which really is the key, that at the end of the day, you want to translate whatever you learn to antemortem so that you can create specific biomarkers. And recently, we just were able to solve the registration based on the initial segmentation between antemortem and the corresponding postmortem. This is a video. I don't know if it worked. But yeah, so just using gradient-based classic optimization, deformable registration, we were able to get one-to-one correspondence. Yeah, and then finally, we are fine-tuning that registration between ex vivo and in vivo using image translation and registration using a deep learning framework. I think that's it. Thank you.